Good evening, everyone. I'm Robin Garrell, the president of the Cuny Graduate Center, and I'm delighted to welcome you to another in our series of public conversations. As a chemist and a leader of a public university, I'm especially excited about this evening's program, which centers on how scientific ideas and discoveries lead to advances in human health and well-being. At the Cuny Graduate Center and the Cuny Advanced Science Research Center, known as the ASRC, we are steadfastly committed to serving the public through research, teaching, and service. Our outstanding scholars are pushing back the frontiers of knowledge, not only within established fields, but also by forging new paths that bring together scholars and methods from multiple disciplines. As a chemist, I know that when people imagine scientists, they often think of solitary individuals in white coats, laboring long hours who, through some combination of effort and luck, make an important eureka discovery. Well, that may have been true in the 19th century, but that's just not the norm today. The extraordinary advances that have been made in the last 50 years in developing new materials for applications such as solar cells and lithium batteries, in developing medical imaging techniques such as MRI and PET scans that enable rapid diagnoses, and, as we'll hear about tonight, in advancing our understanding of human physiology as the foundation for developing new medicines. These have all been made possible through team efforts and through collaborations involving scientists trained in many disciplines, including chemistry, physics, pharmacology, medicine, public health, and many more. Tonight, two experts, Dr. Kevin Gardner, the founding director of the Structural Biology Initiative at the ASRC and a distinguished member of its faculty, and Dr. Michael Brown, a groundbreaking scientist and Nobel laureate, will help us understand how a scientific idea is transformed into a medical innovation. It's an honor to host this conversation at the Graduate Center. And now I'm pleased to introduce Professor Joshua Brumberg. He's the Dean for the Sciences at the Cuny Graduate Center and also the interim director of the ASRC. He will introduce this evening's speakers. Josh?
Thank you, Robin. It's really my pleasure to welcome everyone to tonight's City of Science event focusing on demystifying drug discoveries. The Graduate Center and its associated Advanced Science Research Center are committed to the idea that our scientific research has a role in advancing the public good. To that end, it is my pleasure to introduce tonight's panel. First, a native of Brooklyn, Dr. Michael S. Brown is the Paul J. Thomas Professor of Molecular Genetics and Director of the Jonsson Center for Molecular Genetics at the University of Texas Southwestern Medical School. He received his medical degree at the University of Pennsylvania School of Medicine, followed by subsequent clinical and research training at Massachusetts General Hospital in Boston and the National Institutes of Health. His research on how our bodies sense and respond to cholesterol levels opened a new field of science and laid the groundwork for the drugs called statins. Designed to lower levels of the bad cholesterol, LDL, and prevent heart attacks, statins are taken daily by more than 200 million people worldwide. With his longtime collaborator, Dr. Joseph L. Goldstein, Dr. Brown has shared many awards for this work, including the United States National Medal of Science and in 1985, the Nobel Prize for Medicine or Physiology. He served for 16 years on the board of directors of Pfizer, and he is currently a director of Regeneron Pharmaceuticals. In conversation with Dr. Brown will be Kevin Gardner, who, as Robin mentioned, is the founding director of the ASRC's Structural Biology Initiative. Dr. Gardner received his doctorate in molecular biophysics and biochemistry from Yale University. He and Dr. Brown were colleagues at the University of Texas Southwestern Medical Center prior to Dr. Gardner joining Cuny in 2013. An international leader in his field, he is now exploring the development of new anti-cancer therapies. At Cuny, where he has leadership, has profound positive impact. Dr. Gardner is the Einstein Professor of Chemistry and Biochemistry at City College and a member of our graduate programs in biochemistry, chemistry, and biology at the Graduate Center. He has been involved in founding several biotech companies, and he serves on the Advisory Council for LifeSci New York City, which has helped direct to our city over $1 billion to improve the local biotech environment. His honors include a Searle Scholars Award, a Gordon Research Chairs Hall of Fame designation, as well as awards in teaching and mentorship from the University of Texas Southwestern Medical Center. Finally, a few words about how tonight's events will unfold. Kevin will lead the discussion with Michael, and towards the end of the hour, Kevin will invite you to submit your questions via the Q&A on the bottom of your Zoom window, and he will do his best to get to as many questions as possible. So I, like you, will now sit back and enjoy the conversation between Kevin and Michael. Thank you, Kevin.
Thank you, Josh. And thanks to all of you who are joining Mike and me for this discussion today, which is focused on something I think that's very timely: how new drugs, new therapeutics come into being. This is an obvious issue for everything from global pandemics, as we've been riding through for the past 2 years, to long recognized issues of chronic and acute disease. In general, as Robin noted, this is a team exercise. It requires teams of scientists and physicians to work together. But in general, there are two philosophies that have arisen with the kinds of teams that are put together. One is more disease focused, assembled in its first point with a focus on combating a disease or class of diseases, then very systematically trying to go after that. A second and alternative approach is one that's based more on general curiosity driven research, tackling basic issues, fundamental issues in how life works. And from those, understandings are generated that can lead to cures for diseases that sometimes come sometime later, but can often be more general in their approach. We're going to spend the next 35 minutes or so going through a couple of case studies in this area, and then talk about general discussions of organizations and ideas in these approaches between these two different ways to tackle finding new therapies. With that, I'd like to get started. Mike, very glad to be joining you today virtually in the Zoom world. Let's start with some work very near and dear to you. Let's talk about the Ldl receptor and how the work that you and Joe did led to both basic understanding of cholesterol metabolism and then eventually into the statins. Let's do the first part first, Ldl receptor and basics of what it's there for, what it does, why it's important to understand.
Well, thank you, Kevin, and it's nice to be back with you as our former colleague here in Dallas moved up to the Big Apple, but it's good to see you again. So the story of our work begins at the NIH, and it involves two people who, this other fellow, Joe Goldstein, and I have worked together for over 50 years, which is, we think we have the longest scientific partnership in history in any field of science. But we met as residents at the Massachusetts General Hospital, and then we went to the NIH. And we were both trained as physicians. We had both graduated medical school, but we hadn't done any serious research. And when we were doing our fellowships at the National Institutes of Health, we saw a little 6-year-old girl who was hospitalized at the NIH because she was having heart attacks. The coronary arteries that carry blood to the heart muscle were being blocked by deposits of cholesterol. Now, normally we see that in 60-year-old men, but there was a 6-year-old girl and her 8-year-old brother who both had several heart attacks and were in fact quite ill from coronary heart disease. And the reason was that the level of cholesterol in their blood was 10 times above normal. And at the time, nobody really knew anything about how the level of cholesterol in the blood was controlled. And we have to remember that cholesterol as a substance, as a chemical, was discovered in the late 19th century because it was isolated from gallstones. Gallstones are made of cholesterol. And the structure of it was worked out in an absolutely beautiful way. Cholesterol is a very complicated molecule. It has 4 rings, and it has 27 carbon atoms, and they're arranged in 4 rings. And so working out the structure was a big deal, including the first crystallography of a small molecule. And then the question was, well, how does nature assemble such a molecule? And these very, very fundamental basic scientists, brilliant ones, a man named Conrad Bloch at Harvard and Fyodor Lenin in Germany and others worked out the chemical pathway by which cholesterol is made. And it turns out that all animals make cholesterol, and they need it because it's part of the membrane that surrounds every cell in the body. But consistent with the theme of this evening, the people who worked all that out had no knowledge that it had anything to do with heart disease. They certainly didn't know that it was fundamentally involved in cancer or even in infectious diseases. They really had very little idea of what its actual function was. It was just an intriguing problem. They were driven by the challenge of figuring out how nature could make such a molecule. And I respect these pioneers tremendously. And it's an illustration because without their work, nothing else, none of this could have possibly happened afterwards.
So in that regard, actually, when was the link between high levels of cholesterol and cardiac disease? When did that begin getting appreciated?
Well, it began, well, the human evidence, there was evidence in animals that if you fed them cholesterol, especially rabbits would develop heart disease. But the human evidence began to be collected in the 1950s. After World War II, when people began to do epidemiologic studies and find out, and they measured cholesterol in the blood, they measured cholesterol in the diet, and they found that heart attacks went up as the cholesterol in the diet went up and as cholesterol in the blood went up. And then they found out that cholesterol doesn't just float around in the bloodstream. Cholesterol doesn't dissolve in water. It has a 0 solubility in water. And so if cholesterol is and yet cholesterol is essential for life, for every cell. So how is it distributed? How is it carried around from cell to cell? Well, in the 1960s, it was discovered that cholesterol is packaged together with proteins in particles called lipoproteins. And one of the lipoproteins is called LDL, low density lipoprotein, which the layman thinks of as the bad cholesterol. Well, LDL particles are little spheres about the size of a virus, and each one contains 1,500 molecules of cholesterol surrounded by a detergent coat of phospholipids. And so let's go back to this 6-year-old girl. The reason her cholesterol was so high in her blood was because she had a 10-fold increase in the number of these little LDL globules in her blood. But nobody knew anything about how LDL was controlled. And as physicians, we were very frustrated because there were no medicines for cholesterol back in the 1960s when we saw her. The coronary bypass operations weren't being done. Heart transplants were not being done. There was almost no way to treat these children. We knew it was genetic because her brother had the same condition. We knew it had nothing to do with the diet because we put them on 0 cholesterol diets, she and her brother, and the cholesterol didn't go down. So we knew it was some gene that was defective. So that's how we got started.
So then how do you even start tackling something like this, right? You've got this, like you said, the smoking gun is there. It looks from her case and her brother's, there's clearly some genetic defect. Many of the tools that I take for granted as a scientist candidly born about this time, the tools that I take for granted now were not in existence. How did you and Joe start parsing this out?
Well, the novel thing that we did was to study the problem in tissue culture cells. So right at the time we began these studies in the late 1960s, other scientists figured out how to take human cells and actually grow them in dishes, in what we call petri dishes. And those cells are living cells. And it turns out that they make their own cholesterol. So human cells have the ability to make all their own cholesterol, and they grow. And we decided that I should point out that the condition in these children is a very rare. Only 1 in a million children has this genetic disease. It's called familial hypercholesterolemia. And so we knew we couldn't really learn anything by studying the children themselves. But if we took their cells in tissue culture, maybe somehow we could figure out what was wrong. And to make a long story short, what we found was that when you grew cells in tissue culture, you always grow them in serum, in blood plasma. And the serum contains these LDL particles. And the cells need cholesterol. So it turns out that the cells actually use the cholesterol that's in these LDL particles. And the way they use it is they produce a protein on the surface called the Ldl receptor that actually attaches to the LDL that's in the medium surrounding the cells, and it takes it in, and the receptor takes the LDL into the cell and releases the cholesterol so that it can be used by the cell for its own purposes. So normal cells in tissue culture would use the cholesterol in LDL. When we got cells from one of these children with hypercholesterolemia, we found out that they could not use the cholesterol in LDL. They lacked these receptors. So that now the cells survived in culture because they turned on their own ability to make cholesterol, and but they couldn't take up the LDL from the medium. So then we found that in the body, these LDL receptors are found mostly in the liver. And so our bodies produce these LDL particles, and then the liver uses receptors to take the LDL out of the circulation. And in the children, the LDL receptor can't be made because they have mutations in the LDL receptor gene. And as a result, the LDL builds up because it can't be taken out of the circulation. It's not filtered out, and it builds up, and eventually the cholesterol deposits in their arteries and produces heart attacks. So that was the fundamental discovery. And it was not only that we discovered the receptor, but the whole idea that a cell could produce such a receptor and use the receptor to choose what to eat. That was totally novel at the time. And but then it turns out that cells make other receptors that are specialized for taking up other things, such as iron and vitamins and even hormones. And so it became a general principle. And I think that's one reason why our work was of importance. So it was done at a fundamental level. Then the question is, well, what can we do with this information? Can we use this information? Well, let me go back one step. What about all the other people? What about the 60-year-old man who has a heart attack? Mm-hmm. If he has normal LDL receptors, why is his LDL level high enough to cause him to have a heart attack? Why doesn't the LDL receptor remove all the LDL from the blood and keep his LDL level low? And what we found was that if you look at animals as experimental animals, if you feed an animal cholesterol, the cholesterol goes into the liver And the liver decides, I don't need any more cholesterol. So the liver reduces its production of LDL receptors. And so when we eat these high fat, high cholesterol diets, our LDL levels go up because the liver produces fewer LDL receptors and the LDL builds up until it gets into the danger zone for heart attacks.
So taking away both the 6-year-old and the 60-year-old cases, it's clear LDL receptor is essential in regulating cholesterol, LDL levels, more precisely, in the circulation, either due to a genetic defect, the 6-year-old case, or in just down regulation from just exposure to 60 years of a diet.
Right.
How can this then be leveraged? And agreed then, this is a very general finding in many, many ways for receptor-mediated endocytosis and signaling for many different things, to cells know allowing cells to know what's going on in the world around them and to react accordingly.
Yeah.
How then could this be leveraged into a drug? How did this then initiate thinking towards something like the statins?
Well, this is where something very important happened in science. Science is nasty 4-letter word called luck. Okay. If any scientist tells you, and Kevin, you know this as much as anyone,
Yes.
if any scientist tells you that luck played no role in their major discoveries, that science is not telling the truth. Okay. In our case, luck came about in the shape of a Japanese scientist named Akira Endo. He was working at the Sankyo Drug Company in Tokyo, and he was searching for small molecules, natural products that would block the key enzyme in the production of cholesterol. And Endo found a molecule that would block the production of cholesterol, and that turned out to be the first statin. And Endo didn't know anything. He had read our work about LDL receptors, but he didn't put the two things together. And so we contacted him and we collaborated with him. And we showed in tissue culture cells that even though cells have LDL receptors and they can take it up from the outside, they also keep producing cholesterol. So the cells really have two sources of cholesterol. And if you add this statin drug, you block the production. That lowers the cell's content of cholesterol, and the cell responds by making more LDL receptors. And it turns out the same thing happens in the liver. So even though we're eating all these high cholesterol diets, the liver is still producing cholesterol. And we realized that if we could block that production with Endo's statin, then that would lead to an increase in LDL receptors in the liver, and the LDL level would fall.
And two things I think are important here for the audience members: when you're describing found a small molecule, this is not like walking into a library and just picking random bottles and shaking them on cells, right? This is a natural product that came out of a mold or
penicillin mold. It's a kind of penicillin mold, and so it's very interesting that nature has provided us with these things if we're smart enough to find them. But he claimed he had screened 2,000 different strains of fungi until he found one that produced this compound. So we realized that this might be good for treating the average person who has a high cholesterol due to diet. And so we went to the Merck company, and we had known the CEO of Merck, named Roy Vagelos, for a very long time. And so we helped them, and they started their own program, and they found another mold that made a slightly different version. So Merck ended up with its own statin. and Merck, I give them tremendous credit for developing this, because there was a lot of concern in the beginning. Well, cholesterol is such an important fundamental thing in nature. Every cell needs it. And here you're giving a drug that blocks cholesterol production. Isn't that going to be fatal? And the scientists at Merck had the confidence to continue to develop that drug. And in 1987, the first statin was approved by the FDA. And as Joshua said earlier, they're now taken by 200 million people in the world.
You raised a really important point that I want to make certain everyone's quite clear on the timing here. You said that you and Joe started at NIH with this 6-year-old familial hypercholesterolemia case in the 1960s, and then the first statins are hitting the market in 1987, correct?
That's
so
correct.
About a 20-year arc,
right,
requiring a bit of good luck, requiring people on opposite sides of the world in very different areas of science and medicine to be able to work together and contact. Yeah, actually, I'm curious. One thing I actually don't know, who contacted who between you and Joe and Endo? How did that initial contact get made?
Fascinating story. It turns out that Joe subscribes you remember, this was before Google, okay, and before the internet. But there was a computer-based service that you could pay money to them, and they would send you a list of every article that was published that referred to one of your papers. So you would get these huge computer printouts on this perforated paper every week. And Joe's glancing down this, and he sees this paper in the Japanese language from Japan, and it quoted all of our papers. And so we got the paper, and we got one of our Japanese colleagues to translate it. And we saw that he had read all of our work, and it was a review article based on our work. So then we set up a system where we followed his publications. And the next year he published his paper showing that he had this statin. And so we contacted him, and he happened to be coming to the US to a scientific meeting. So we invited him to Dallas, and we started the collaboration. But if we have time, there's a side story. That same molecule had been discovered by scientists in England at the Beecham Pharmaceutical Company. They had found it because they were screening fungal extracts for antibacterial drugs, and it came up in a screen, and they purified it, and they determined the structure of the molecule. They had no idea that it had anything to do. First of all, it was a very weak antibacterial. Bacteria don't have cholesterol, so it had nothing to do with that. So they discarded it, but they published a paper with the structure. So, and so they thought that it was like a weak antibacterial, but when they read Endo's paper, they took some of their drug and they fed it to rats, and they told us, oh, it's terrible. It doesn't lower the cholesterol at all. And they sent us kilograms of it. I mean, we had the world's largest supply because the Beecham company wasn't interested in it anymore. It didn't lower the cholesterol in rats. Well, they didn't realize rats don't have LDL in their circulation. Rats use a totally different lipoprotein to carry their cholesterol. And so that you wouldn't expect it to lower the cholesterol in rats. And it's just a piece of knowledge that they didn't appreciate. So it just shows you how sometimes discoveries can slip right past you.
Again, luck playing a role in discovery, in this case, bad luck,
Exactly.
to not fully appreciate the differences.
Well, it's just not doing your homework and reading about rats. But so it's really the courage of Merck that and the Merck executives that pushed this forward, and then it turned out they did the largest safety study that had ever been done at that time, and it was totally safe. And you know, I can't go into all the details. Yes, statins do have side effects. Some
Mhm.
people have muscle aches. And if you're a borderline diabetic, it can sometimes raise your blood sugar enough to give you, be classified as having diabetes. And those are serious in some people, but the vast, vast majority of patients benefit from statin drugs.
And again, based on a foundation of work that had occurred well in advance of therapeutics being even brought into the equation, right? You're interested in the molecular root of disease.
Exactly.
And then that together with what was coming in a very parallel track, back and forth catalyzed by luck.
But remember, the original scientists who didn't know anything about heart disease, they just thought
Yeah.
cholesterol was an interesting molecule. And you know, and if there was no support for that kind, if there is no support for that kind of research, then the next time we have something, there won't be that fund of knowledge around to draw upon.
Absolutely. And if we could transition here a little bit into the next time something comes along department, thinking back to some work, pioneering work out of your alma mater at Penn, with what we've been dealing with the COVID-19 pandemic. Could you give, I think people on the call are likely to be vaguely aware of, well, they've heard about these wonderful RNA vaccines, but and how these came seemingly out of nowhere for many people, right? One year between less than a year between sequencing and
Right.
vaccines being put in the arms of millions of people worldwide. But that wasn't just a year's worth of research. That was sitting on top of a long, long story before. Could you give a brief vignette of
Well, I mean, it goes
what
back,
came before all that?
you know, it goes back to, you have to realize that the ability to make an mRNA vaccine, first of all, you have to know about messenger RNA. And to know about messenger RNA, you have to know about DNA. And then to know about DNA, you go back to Watson and Crick and a famous New York scientist named Chargaff. But the fact is that once the structure of DNA was discovered by Watson and Crick, and it was known that DNA could produce proteins, people had to figure out how is it translated from the DNA sequence into a protein, which is a sequence of amino acids. And so, and then these brilliant scientists discovered messenger RNA, many of them from the MRC in Cambridge. So the fact of the matter is, and none of them was concerned about disease. Again, they were simply trying to figure out how nature worked. You know, how does heredity work? And so that started it. And then you had the advent of something that I mentioned earlier, which is the ability to take cells in tissue culture, and the people had to figure out how to grow cells outside of the human body. And then how do you get this messenger RNA into a cell in order to get it to work and produce proteins? All of that stuff had been built up over all of these years. But there was one problem, and that is that when you injected messenger RNA into an animal, it turned out to activate the immune system. So in order to understand that, you had to know an awful lot about the immune system. And the people who discovered all of that were not specifically looking at viruses. They just wanted to figure out how the body reacts to foreign invaders. And so, but these two brilliant scientists at Penn who had Katalin Karikó and Drew Weissman, and Karikó has the most interesting story. She was an immigrant from Hungary who had a terrible time getting support for her research and basically worked as an indentured servant in other people's laboratories because she didn't have independent research support. But she figured out how to modify RNA so that it didn't cause this inflammatory reaction. And Weissman was an immunologist, and he knew that you could use this to make a vaccine. And out of that vaccines were born. So I look at basic research as putting books on the shelves in a library. You never know when you're going to need that book, but when some new virus comes along, or people start dying of heart attacks or cancer or Alzheimer's disease, you can, if you've done the basic work, you just go to the library and pick up that book. And now I realize that libraries are, a lot of people never heard of a library anymore, but think of it as Google. The fact is, once you have that information, okay, it's there, and it doesn't matter how it got there. It's and who and what the aim of the person is. And I'm a little bit afraid now, Kevin, that right now there's so much pressure on the research agencies in America to produce results. People want a cure for Alzheimer's today, not, my mother is getting old enough and I'm worried that she may have Alzheimer's and we need a cure today. And it turns out that you can't just cure. So if the basic knowledge is not there, you can't do it, no matter how smart you are or how much money you pour down the drain. And I one example I use is sickle cell disease, a horrible, horrible disease that affects a large number of African Americans. There has been so many requests for proposals and money poured into finding a cure for sickle cell disease, and nothing happened until the basic science caught up and people figured out how to change genes, and suddenly now there may be a cure for sickle cell disease. Certainly there are clinical trials that are showing tremendous results since we learned how to manipulate genes. So you can't get ahead of the basic science. If you don't have the basic science, if you try to imagine a cure for something and you don't have the basic knowledge, you're just going to go off in crazy directions. And
they're the molecular basis of the disease, right? A single point mutation in one of the changes of hemoglobin has been known since the 1960s.
It was the first genetic, right? It was the first genetic disease where a mutation was identified.
So let's talk for a few minutes about how you can build a good team to be able to get that foundation, the critical foundation of basic science together. I mean, there are challenges, and I already see one question in the Q&A, and this is a good reminder. Please, we'll be taking questions and answers at the end of our conversation, roughly in about 10 minutes or so. If audience members, if you have something that you'd like us to explicitly talk about, please put it in the Q&A box below. But Mike, you and I come from very different traditions of formal learning. As a medical doctor yourself, I'm a PhD biophysicist. These are, for people on call who aren't in biomedical research, we're in quite radically different worlds.
Yeah.
But we find ways to be able to talk. And to you, how do you get the right people together and critically not just have them kind of stand isolated in their own little worlds, but get them to start interacting with each other and sharing what they know and how you might be able to tackle a big problem like sickle cell?
Well, Joe and I have each summer we take summer students, high school and college students, and then at the end of their experience in our laboratory, I ask them what the biggest surprise was, and they always say, "I had no idea that science took so many people, and that you guys work in teams." Because that's not the way it's taught. The average student thinks a scientist gets a brilliant idea, goes into a trance, does an experiment, and goes to Stockholm. The fact of the matter is that there's a lot of hard work involved. And nowadays, there are many disciplines that have to come together, people's training in different areas. So for example, you're a physical chemist and your knowledge of protein structure can provide the answer to a question that I have about a certain kind of cancer But somebody's got to bring us together and let us talk to each other. And so I think it's really important to have centers of excellence where you bring people with different backgrounds, scientific information, bring them together, but they've got to interact intimately. You can't just do it, by sending emails. It really does require constant dialogue, and so yes, that the answer may be there, but it's in somebody else's head, and we've got to unlock it. And here's an example. A friend of mine, a very prominent physician in Boston, was teaching a class at MIT to engineering students. And he told them that one of the big problems in medicine is that elderly people fall, they land on their hip and they fracture their hip. And then frequently, that can lead to death in an elderly person. He asked the engineers, what would you do about this? And these engineering students thought about it. And then a group of them came to him the next day and said, we have the answer. Old people should wear airbags. And as they fall, these airbags inflate around their hips and protect their hips. There's no way a physician ever would have thought of that answer. If you would ask any physician, how are we going to, they would talk about bone growth factors and, giving people more calcium. But it took the way engineers think. So bringing engineers together with biologists is also extremely important.
Actually, I'd like to then pull on one other aspect of broadening who gets involved in biomedical research. I think you've hit one important area, which are getting people together from very different disciplines. Also, take a minute here to note that, especially within a system like Cuny, we're really a big focus of our organization. I think many other increasingly across the US are getting a broader group of people, especially traditionally underrepresented groups, women and people of color engaged in biomedical research. We're real proud in Cuny, for example, of a woman by the name of Marie Daly. She's a graduate of one of the Cuny colleges, Queens College, goes on to Columbia to become the first Black woman to get a PhD in chemistry anywhere in the United States, and goes on to have a great research career, which actually does touch a little bit on the link between hypertension and cholesterol biosynthesis. But I think if we're truly going to hit the grand challenges for biomedical research of the next decade and beyond, from where I sit, we need all hands on deck, right? It is whoever, I want the best and brightest minds to be engaged in the problem, engaged in the act of discovery, engaged in getting new cures. And anyone who's left on the sidelines because their skin's not the right color, they're not the right gender, they're different in some way, shape, or form, the time for that is long past gone. I think you've hit on one really important thing there, summer students in early engagement, right? I wish Hollywood would show biomedical research as it is. Hands-on experience is a real good way to help address that. Other ideas or other thoughts that you've seen work well or that you practice in your own group with Joe?
Well, I certainly agree that we don't have a brain to waste, okay? There are too many problems, and we need every brain that we can put on these problems. And the brains come from all over. It's not just the image that we have maybe of you and me, and all scientists should look like us, with the exception that I'm old and you're not. But the fact is that I was thinking the other day about the mRNA vaccines, and there's a thing that is just absolutely striking when you think about it, okay? Because the Pfizer vaccine was created by a woman, Katherine Jonsson, who as Pfizer's head of vaccines, who emigrated to the United States from Germany. And she was supported in this whole effort by the Pfizer CEO, Albert Bourla, who came to the US from Greece. And then the fundamental work on the mRNA was done by Katalin Karikó, who came to the US from Hungary and worked together with an American, Drew Weissman. And the original Pfizer vaccine was the pioneering work was done in Germany by two Turks, Ugur Sahin and Özlem Torici, who are married to each other, one a man and one a woman. And they founded this company called BioNTech. That's why it's called the Pfizer-BioNTech vaccine. And even Moderna, the CEO of Moderna is Stéphane Bancel from France, okay? So look what it took to make those vaccines. It took all these brains coming from different places, half of them female, and that just shows you, what's going on. So, we have a lot of Chinese students, and they say, China keeps telling us that they're going to bury us because they have 1.4 billion people and we only have 350 million. And I say, we don't only have 350 million. We actually have 4 billion people. If we can get the best and the brightest from the world to come here and work with us, then we have 4 billion. And that's what has made America great from the very beginning, giving opportunity to everyone. And as you point out, there have been some overlooked, obviously horribly overlooked groups that we haven't given the opportunity to, and we're correcting that. And places like Cuny are avenues, traditional avenues that offer this opportunity. And you say, well, how do we start? And I think it all starts with education, and the earlier you can stimulate people to think about science, the better. And we have problems. A few years ago, I gave the commencement speech for the College of the University of Pennsylvania, and there were 10 students who were getting honors at commencement. The one getting the best was the best physics student, the best chemist, the best biologist, the best humanities student. And I asked each one of them what they were going to do next year. And I swear to you, every one of them said, I'm going to work on Wall Street. Now, I have nothing against Wall Street. I own a few stocks myself. But the fact of the matter is that we have to get, now the problem with it was that the year that this happened was 2007, right before the enormous crash of 2008. So I don't know how long these people stayed on Wall Street, and hopefully some of them went back to graduate school. But the fact is, we need to compete for these brains. That we as scientists and engineers, we need to compete for these brains.
Someone's asking the question over on the Q&A, and we'll transition there in a minute, but it's exactly on this point. How do we get this young generation of students excited about something like this? I mean, for me, given, I'm of a certain age, that I remember sitting in front of a TV set and watching 3 astronauts at the top of a really tall bunch of really explosive fuel get shot into the sky. And while I think there are legitimate discussions one could have now about return on investment for what you learned from some of that, from that investment in the national enterprise, boy, I will say it was a very powerful motivator for a young version of me to want to go learn more about science and technology. How can we get more students excited about this? How do we go get them engaged on things like this?
Well, first of all, you bring up the moon program. the fact is that that was all started by Sputnik. I mean, I was in college when Sputnik happened, and the U.S. felt horribly threatened. And John Kennedy said, "We're going to send a man to the moon and bring him back alive," and within 10 years. And that was a huge motivator for students in science to go into science. And science became really popular in the early 1960s, and graduate schools expanded like mad, and a lot of people went to graduate school in physics. Then came the Vietnam War, and the Vietnam War caused all this distrust of science. Oh, scientists are the ones who made Agent Orange and dioxin and all these bombs, and scientists are evil. And so there was a reaction against science. I think now we should feel just as challenged, at least as challenged as we did during the Sputnik era. We're challenged not by hopefully not by atomic warfare, but we're challenged by what's happening to our environment. We're challenged what's happening to the world. And again, we haven't seen the last of these pandemics because of transportation, and has made these things spread so dramatically and rapidly. So we're very threatened, and we need to put more emphasis on science and getting young people motivated. We've got to train our teachers, especially at the elementary school level, in biology. If you just teach children to memorize words, the parts of a cell and all the things they teach in such a dry way, no wonder the students get turned off to science. You have to inspire them with the mystery of it all. And so at any rate, I just hope that that the American society is so fractured right now, but hopefully we can all get together and realize that we're all in this as one, and we're either going to all live together or die.
Yep. Let's see. I'm looking through the questions and answers, and there are 3 that I'd really like us to get into. So I'm going to read those out and encourage some focus answers them as best we can, even though a couple of them are going to hit on some big world topics. So first question I want to get into, put forward by Daniel. Let's talk about the high cost of drugs. Why, given everything we've just been through on the amazing aspects of discovery, why, in a nutshell, very big topic, Daniel, but Mike, why are drugs so darn expensive?
Well, first of all, I have to say that I'm on the board of a pharmaceutical company called Regeneron in New York State, actually headquartered in Westchester. And I was on the board of Pfizer, headquartered in Manhattan. So in a way, I'm contaminated. But the fact of the matter is, what we really need is a serious investigation of all the things that go into drug prices. How much money is being spent on research? How much money are the companies spending on marketing and sales? Are marketing and sales really that necessary? For a long time, television advertising of drugs was banned.
Forbidden, absolutely.
And then it was an act of Congress that allowed direct-to-consumer advertising of drugs. And if you turn on your television, within a half hour, you're going to hear a drug advertisement. That costs money. And so we need to know how the drug companies are actually spending their money. But it's not only the drug companies. There are things called pharmacy benefit managers, or called PBMs. So the drug companies sell their drug to these distributors, which are called PBMs. And the PBMs, for complicated reasons, have an incentive to have a very high list price for the drugs, so they can then offer discounts to the insurance companies that are paying for the drug. That helps the insurance companies and the insured, but it leaves the uninsured person totally out of it. So we have to go beyond just having a 12-year-old boy stand up and say, you know, my parents can't afford my insulin. We need to have, I would hope that the Congress or some investigative body would do a serious and a non-political investigation of the whole drug industry. How are they spending their money? And you have to realize also that you have to have profits. That has to be part of the equation, because it's the profit motive that is driving the venture capitalists to invest money in things that are probably won't work.
And probably won't work. I mean, that's an entire other hour I think we could get into. The odds against an idea turning into something that even begins a clinical trial are daunting, much less than completing the clinical trials successfully and off to where we are now. Let me touch on two other topics from the Q&A very briefly, just before we need to wrap up. So Giannis asked a question that is, I think, really forefront in my mind is, boy, in 2022, in the public arena, science is not where I think any of us, I suspect, on this call would like it to be. It is, things even as trivial as whether someone's wearing a mask or not in public is viewed as a political statement rather than a wise public health decision. How in the name of blazes do we, what can we possibly do to try and jumpstart this and get us back to a little bit more, putting my cards on the table, a saner view of the world, and where science can answer the questions it can answer? Thoughts?
Well, I think the problem is deeper. It goes beyond science. It's just a trust in authority. It's we've lost our trust in the rules that govern our country, and it's if I weren't, I'm at a certain age that I really don't have to worry about what's going to happen in 2030 or whatever, but I have grandchildren, and once you have grandchildren, suddenly this is all very meaningful. I look at my little 12 and 9 and 8-year-old grandchildren and say, what is the world going to be like for them? Are they going to have the kinds of opportunities that I had, and are they going to be able to drink the water? Are they going to are the cities going to be inundated? Is this whole, my 8-year-old just wrote a paper last night on the plastic. There's apparently a sea of plastic garbage somewhere in the Atlantic Ocean, the size of Texas. And it's just a sea of plastic garbage. And, at least he wrote about it. But, there's so many things with this planet that need addressing. And just when we're questioning all authority, it's a bad combination.
One can only hope that some of those at least can be a good motivator, like the Apollo moment was for me, and I think many other people in my generation. I hope that your grandson and other kids see that and get a little bit upset about it and get curious as to what they could do to actually try moving things forward. And same thing for the little young boys and girls on the call, or their parents or grandparents who are on the call today, for those kids who want to make contributions over in biomedical science. I know, Mike, I think you and I both have very much enjoyed our careers in this area, and all the different ways it's taken us in directions we might not have guessed, especially when we first got into it.
No, I went to medical school because I wanted to be a doctor, a physician. That was my motivation. The science came later. And it was all due to exposure, in one laboratory, exposure with one great scientist. I think, if any student gets an opportunity to work with a scientist who's really authentic, and because not all scientists are great role models, but to work with a scientist who really is at the forefront and is critical and is trying to do things, learn new things and not just repeat something somebody else did. You've got to find scientists like that and try to get exposed to them, and then great things can happen.
Hear, hear. I think that's a wonderful point to wrap up for tonight. To the many people who have asked questions over in the Q&A that we haven't been able to get to, I sincerely apologize. I've tried folding some of those topics into some of our discussion here this evening. I love where that's going, and maybe with support from Josh, President Garrell, and the GC, we can all get back together again another time, preferably in person even, to continue this. Mike, thanks again so much for your time. Audience members, thank you for being part of a public discussion at the Graduate Center on the important influence of science in our society. Thank you very much for your time, and have a good evening.
Thank you, Kevin.
Thank you, Mike.